Assalamualaikum warahmatullahi wabarakatuh. The first, I want to say thank you to this committee to give me opportunity to present our research, with the title is Polymorphism of Follicle-Stimulating Hormone Beta Gene as a Molecular Marker for Reproductive Status in Poba Cattle. I am Almira and this is our team, Mr. Johari Effendi and Dr. Penny Wahyu Prihandini from Beef Cattle Research Institute, the Ministry of Agriculture Indonesia. I would like to start with introduction. The Poba cattle are crossbreed cattle from Peranakan Ongol male and Bali female cattle, which are developed by the Beef Cattle Research Institute in 2015. In 2018, Poba cattle consists 2 bulls and 10 females. Filial 1 were made to produce Filial 2 with natural mating was applied with a ratio of 1:5. The population of Poba cattle at the Beef Cattle Research Institute in 2019 was 24 heads. There is no pregnant cattle based on a pregnancy test in the 4th month after being collected in the mating cage. That's why it is important to conduct a study in order to determine the reproductive performance of Poba cattle, both phenotype and genotype traits. The phenotypic performance of superior cattle does not guarantee their reproductive superiority, and thus the use of molecular markers as a selection tool may be useful to increase the accuracy and efficiency of traditional selection, especially for reproductive traits. Therefore, it is imperative to conduct research exploring genes controlling reproductive traits in Poba cattle, such as follicle-stimulating hormone subunit beta or FSH beta gene. FSH is a glycoprotein hormone secreted by the pituitary gland and has a function to control reproductive activity in mammals. FSH can stimulate the growth of ovarian follicle as well as initiates and improve the activity of Sertoli cells, the process of spermatogenesis. Polymorphism in exon 3 of the FSH beta gene resulting 3 alleles, A, B, and C, significantly affect the volume, quality, and motility of both liquid and frozen semen in Limousin, Hereford, and Friesian Holstein cattle. Significant association between FSH gene polymorphism and superovulation has been identified in Chinese cattle by Yang et al. Polymorphism in the FSH beta gene is detected in Brahman, Friesian Holstein, Simmental, and Limousin cattle. However, the gene is found to be monomorphic in Bali cattle. The materials and methods were used are blood sample collection from Poba cattle. In total, 11 Rambon cattle collected from Banyuwangi Regency and 24 Poba cattle collected from Beef Cattle Research Institute. The primer used following Dai et al. 2009 and Isaac et al. 2011. The analysis used in this study is sequence or SNP analysis using bioedit software and manual detection to the electroperogram. Calculation of allele and genotype frequency following formula Naik and Kumar, Harjo Subroto, and Nur for Hardy-Weinberg equilibrium analysis. The 313 of the the 313 base pair of the FSH beta gene sequence were successfully amplified for all samples from Rambon and Robacetal. The result of the PCR amplification, like Dai et al. 2009 and Isaac et al. 2011. The polymorphism of the FSH beta gene in Rambon and Robacetal was analyzed using the sequencing method. The sequencing results from all samples were aligned with a reference sequence from GenBank. Three genotypes, namely TT, CC, and CT, were observed. In the table 1 shows the allele and genotype frequency for SNP of the FSH beta gene in Rambon and Robacetal. TT genotype is higher than CT and CC genotypes when T allele frequency higher than C allele in Rambon and Poba sample analyzed. There were two alleles, C and T, in Rambon and Poba cattle, which indicated an absence of selection in the both cattle population. Table 2 shows a Hardy-Weinberg equilibrium for SNP of the FSH beta gene in Rambon and Poba cattle. Only Rambon cattle were in Hardy-Weinberg equilibrium, while Poba cattle showed a deviation from Hardy-Weinberg equilibrium. This deviation may indicate a negative selection that can lead to inbreeding in the population. Therefore, new bulls are needed to be introduced. In Table 3, show the observed and expected heterozygosity for SNP of the FSH beta gene in Rambon and Poba cattle. The heterozygosity value indicated the level of crossbreeding in the population. Association between genotype and phenotype shown in Table 4. The association between genotype and sperm quality in Poba cattle. Poba male cattle had poor sperm quality because the sperm was not found during the storage process. Therefore, there was no value for motility, mass movement, and concentration. There was no association between FSH beta genotype and sperm quality in Poba cattle. The Table 5 shows the result of genotype and phenotype, body weight, body size, and cervical condition of Poba cattle. There was no significant association between FSH beta genotype and body weight, body size, and cervical condition. So the conclusion for this research is there was a polymorphism in the FSH beta gene in Poba cattle indicated by the presence of three genotypes, TT, CC, and CT, with the number of animals with TT genotype is higher than CT and CC genotype. The frequency of each allele was 0.13 for allele C and 0.87 for allele T. The FSH beta genotype had no significant association with sperm quality, body weight, body size, and cervical condition of Poba cattle. That's our presentation. Thank you for the opportunity. Assalamualaikum warahmatullahi wabarakatuh.